I'm so happy to introduce our next speaker. And we're going to, Paul Cox has been a huge supporter of our Center for Chemical Ecology, and he's distinguished in many ways. He was once in Time Magazine Heroes of Medicine for his work. He's an outstanding speaker, outstanding colleague. He studied at Harvard as an NSF fellow, where he received a PhD. He was the dean of the Brigham Young University and a professor in Sweden. I mean, he's got so many stories. I can't do justice that way. He's been recently on CNN, and he's definitely demonstrated overall just how this whole understanding of this chemical world can provide tremendous benefits to us humans, but also narrow in on these wonderful things. So with that, please join me in welcoming Paul Cox.
I'm really glad to be here, and I want to tell you why I think the work that's happening right now at the University of Nevada, Reno, is some of the most important science happening in this entire world. And let me just put that in perspective. In 1912, 1914, if you were interested in physics, you'd better get yourself to Copenhagen and listen to what Niels Bohr had to say, and Heisenberg and those guys developing a new way of doing physics. These folks here at this university are developing a new way of doing biology, which they're calling chemical ecology. Let me just spend a few minutes with you telling you why I think what they're doing is so important. One of the central questions in biology is what maintains the diversity of tropical rainforests. If you go into a hectare of the Amazon and count the number of tree species, there's more trees in 1 hectare, what's that, 2.5 acres of rainforest in the Amazon than you've got in all of New England. Okay? I mean, this is a real conundrum. Everything we find out about the tropics shows us that it's breaking molds. Now, if we go back to the history of biology, Aristotle, and of course his teacher Plato, thought that every species has an eidos, it has a little place it should be. You can write them all down. Theophrastus, Aristotle's student, did that in De Causis Plantarum. And so it persisted until this guy, Carl Linnaeus, who was in Uppsala, Sweden, left in 1732, went up through Lapland. I got to follow his trail. I was a professor over there. And he came up in 1736 with a book that contained all the known plants of the world called Species Plantarum. He carefully organized them. Everybody had their little way of doing things. And that was really great until this guy came along, Alexander Humboldt, about 50 years later. 1799, Humboldt left for a journey into the tropics. And when he hit the rainforest in the Amazon, biology changed forever because there were too many species. There's too many species in the rainforest. I remember the first night I spent in Costa Rica in the rainforest, I had nightmares. I was a botanist, and I was just overwhelmed I saw more species in 1 hour than I'd seen my entire life. What is going on? Why are there so many species? And this is a really important question to answer. In fact, I'd say it's one of the most compelling unsolved questions in biology because the rainforests are the lungs of the earth. They help maintain the atmosphere. They're the source of medicines. They're important to the people that live there. What's going on? Well, a Russian biologist in 1936 named Gauss wrote a book which translates The Struggle for Existence. What Gauss said is that every species has its niche. You can only have as many species as you have niches. And this has been maybe one of the central dogmas of ecology and ecological theory ever since. That every niche has one species. If you had two species that had the same niche, they would fight, they would eventually extinct one and the other would remain. Well, that's gone out. And I want to show you why what's happening. If you want to understand that question right now, that question, in my opinion, is being solved right at the University of Nevada at Reno. Let me show you why I'm saying that. This is Piper kellii in Ecuador. This is a small understory tree. Grows in Ecuador and up in the foothills of the Andes, in the jungles. And this are the species Eos. These are caterpillars. What these guys have found here at UNR Reno is that you can go into that jungle, you can look at the bottom of a leaf, and you can find a gazillion different species of caterpillars all eating the same leaf. This violates everything that Gauss said. There should only be one niche. In fact, they eat it the same way. They scrape the bottom of the leaf. In fact, some of them look alike, but they're different species, and they haven't finished counting up how many species is going on. Well, who are the folks coming up with this stuff? We got Lee Dyer, Andrea Glassmeyer, Laura Richards, Angela Smolanich, Mark Forster, who wears a butterfly in his cheek, Christopher Jeffrey, and a zillion students are all working right now on this puzzle. How can one plant species have underside its leaf so many different species? And their answer is mind-blowing. It has to do with chemistry. They're finding that in this one species, that depending on how high it is in the canopy, what the altitude is, it produces these different chemicals that deter the caterpillars. And that they vary. And this answer that is emerging, they're not quite there yet, but they're very close, in my opinion, requires a zillion different people to answer it. It requires chemists, it requires rainforest biologists, it requires physiologists, it requires entomologists. And what they're finding is that the plant is talking to its herbivores, its predators, with chemistry. And that question, in my opinion, will be solved at this place and this time within the next year or two. And it will change forever what we think about how tropical forests are maintain all these species. It will change the way we manage forests. It will change the way we think about forests. You're seeing the overturning of a century-long dogma that's being trashed but superseded through some really brilliant biology that took an entire different team of scientists. So I love it. If you want to find out about tropical rainforest, where you go to Nevada. And it turns out there's a lot of things you do when you go to Nevada. And this university is going to gain a lot of, I think, just even more prestige than it already has. And, I asked Mick Hitchcock, who founded this whole thing yesterday, or this morning, I guess. I said, God, I hope you're happy, Mick, with what you've done, because you've really unleashed chaos on biological science. But there's so many smart students, there's so many smart postdocs, there's so many smart scientists, all collaborating, all working together, that they can approach big unanswered questions. And that's the promise of chemical ecology. So this brings up the question, how do species communicate with each other? Here we have a cactus, here we have a sheep. So do they vocalize to each other? The sheep goes, bah. And I'm here to eat you is what that means. And the plant can't say anything. It just sort of sits there terrified. Then maybe the sheep counts. You count them to go to sleep. 1, 2, 3, 4, 5. Plants actually count. They use the Fibonacci series as they put their leaves out. But that's not how they communicate either. Boom. How about this? The sheep leaves little souvenirs for the plant. Enjoy some fertilizer. And then, but before I eat you, the plant responds with chemistry. It puts out mescaline, which says, forget you ever came here. So the language of biology is not mathematics. It's not vocalization. It's chemistry. And that, if we understand those chemical interactions, it can lead not only to understanding tropical forests, but some of our most serious problems. This is one of my favorites, Gila monster. I don't know if you follow these things, but some poor guy died in Colorado recently, bit by his own pet Gila monster. I was very sorry for this guy, but his, well, I think I'll leave it there. I'd heard of a graduate student before that got bit by clamping the jaws of a Gila monster on. Gila monsters really don't want to mess with us, but they do need to hunt. And they hunt things like kangaroo mice. So here's how their conversation goes. Please don't eat me. And the Gila monster responds with this. This is a 39 amino acid peptide called exanatide, which immediately talks to the pancreas of the kangaroo mouse and messes up its sugar metabolism so it can get a little better chance to crawl up and slowly grab it. Well, a guy at the VA hospital in New York, and I love this, against the wishes of his superiors, got really interested in this molecule because he found that it hit a receptor in our brains called GLP-1. This is a receptor that controls the release of insulin. It also controls whether we feel full or not. This led to the first major improvement in treatment of type 2 diabetes, a drug called Byetta. Type 2 diabetes afflicts 15%. It costs 15% of every healthcare dollar in America to treat type 2 diabetes. And this led to, you know the song, don't you? Ozempic, yeah. $14 billion a year sale. I understand they're going to start paving the streets of Copenhagen in gold now. Let me give you another example. And by the way, Ozempic, very interesting. Have you heard of Ozempic face? They actually go on the web tonight and look at that. People that are, the poor patients that really need it are fighting for Ozempic from the people that just want to lose a lot of weight. Because boy, hitting that receptor really helps you lose weight. Here's another one I think is pretty interesting. This is called the death scorpion of Palestine. Pretty cool thing. This can actually kill an older person or a child. Jim Olsen up at Hutchinson Cancer Center in Sweden derived this molecule, chlorotoxin, a 36 amino acid. By the way, amino acids are the building blocks of proteins. 36 amino acids long. And it turns out this comes from the venom of the scorpion, and it actually paints cancer cells in the brain. It hits glial cancer cells. So Jim found that if you take a child under brain surgery, paint their brain with this chlorotoxin, and then put on a fluorescent light, you can see exactly where the tumor is. This is a big step forward because, particularly for children, you want to take enough of the cancer, the tumor, to get it out. But if you take too much, they're left impaired. And this has really revolutionized brain surgery. So we're pretty interested in what these guys are doing. By the way, that's one of the patients whose life was saved through chlorotoxin. So we're looking at using chlorotoxin, sort of like the missile, to attack glial cells while we have our warhead. And let me show you that. So this is Dr. Samantha Gerlach in our lab. We're working on cyclotides. These are these weird circular membranes, or circular proteins, that come and you just can't make this stuff up. They come from violets. Here's a Wyoming violet. We were going clear down to islands of Polynesia looking at unusual species of psychotria, discovered that violets make them. It's right in the backyard. This is the one I shot in Wyoming. And what we wanted to do was to see if these would attack glioblastoma, which is a major killer. Most people that have glioblastoma only live 9 months from diagnosis. And it's really bad news for children because, again, surgeons can't pull that tumor out. So we wanted to collect enough of this to see if it hit glioblastoma. And that meant that we were working day and night in Wyoming, collecting violets, extracting them. Extracting these little proteins is quite a process to get just a tiny, tiny bit that we could use in cell culture. And I got frustrated, so I called up one of my friends at National Cancer Institute. I used to work with them a lot. And he says, well, why don't you synthesize it, Paul? Well, I didn't want to tell him that I'm not like Professor Jeffries in my synthetic chemistry class. And I got a good grade, but I can't even boil an egg, Chris. So I'm but we found some people that could do this. And so now here we have that cyclotide that we've partnered with some teams down in Palo Alto to make for us. And now what we're doing, well, let me show you what we found. In the test tube with human glioblastoma cells, it turbocharges the current chemotherapy, which isn't very good, 8-fold. It really makes it massively important. And now what we're doing is we're designing our own artificial molecules by changing some of these amino acids to see if we can find something even more important. Now, we've got a long ways to go before we get to the clinic on this thing because toxicity is going to be an issue. We haven't done animal studies yet, so I have to be very cautious here. But from what we're seeing, this could be a path. It could be a path that would lead to a new way of attacking one of the most serious cancers known. Let me go back to cyanobacteria because you heard Sanjay Gupta, nice guy, by the way, although he tried getting me saying on camera several times, well, wouldn't you take L seriously? I said, no, we know it's safe, but we don't know for sure if it works. We've got to let that clinical trial finish. We just enrolled our 65th patient in Houston, at Houston Methodist, and we'll find out. But the guys down there are pretty excited, the neurologists, what they're seeing, but they're double-blinded, so they tell me this time, I say, yep, that's the power of placebo. And we all have to wait till we see what happens. So cyanobacteria, they're the earliest forms of life on Earth. They first organisms 3.5 billion years ago, something very like current cyanobacteria. I got interested because I knew they created the oxygen atmosphere of the Earth. I was at the University of Melbourne and knew that in Shark Bay in Western Australia there were living stromatolites, so I took my wife Barbara and our little children, flew out there, drove the grueling 12 hours from Perth up to this place, got up there, and all of a sudden I realized I don't know what these stromatolites look like. And I just saw these rounded rocks. I stood and looked around, and it's sort of an embarrassing thing for a biologist to do that, So I went back to Melbourne, said, what do they actually look like? And discovered it was the rounded rocks I was standing on. So I've made some different approaches there, and this is sort of like when the Earth was first formed. Every day the sun comes out, these cyanobacteria just sit and they pump out oxygen, which slowly changes the atmosphere of the Earth to one that we enjoy today. And they had complex chemistry. Here's some stromatolites. So tuck that away. I didn't know what they looked like. It's an embarrassing story.
But let's face it, I'm just a cow pie kicker from Wyoming, and what can I say? The other thing I was going to say, Chris, sometimes I'm introduced as one of the world's top 50 ethnobotanists because there's only 25 of us. So anyway, pleased to lend my support to your organization with paste. So I got worried. I was seeing that the Great Salt Lake was losing water. You know, it's really lost a lot. Lake Mead too. Being Nevada, of course, Lake Mead, water goes down. You find old boats, stolen cars, mafia hits. What does the Great Salt Lake conceal? Well, so I got a helicopter, started going around, and look at that. Stromatolites. Couldn't believe it. They call them microbiolites, I guess, but I'm going to call it stromatolites. Cyanobacterial formations, that's on one of the islands out there. So we got started worrying because we knew that cyanobacteria produce this nerd toxin that we really showed, I think, pretty convincingly was what was killing the people in Guam with this terrible disease. But were they producing the same stuff? So together with one of our senior scientists, a cyanobacterial expert, Dr. James Metcalf, we started going around the Great Salt Lake. And James, a really clever guy, and he has this really beautiful accent. He's English, you know, and the ladies in Jackson just kind of ape over this guy. You know? But a really nice guy. He engineered a device there that is set up at the height that you breathe out, and it collects the same amount of air that you would breathe in an hour. And then we go back to the lab and see what you're breathing coming off the Great Salt Lake. And here, I just had to say, it is so fun to ride in a helicopter. This was really good. So here we are collecting our samples all around. Brought them back to Jackson Hole. And here's something to make Professor Jeffries happy. That's our triple quadrupole mass spectrometer, and Dr. Sandra Bannock, our senior scientist there. We analyzed this stuff, and to our horror, discovered that the dust blowing off the Great Salt Lake towards a population of 2.5 million people has in it that old friendly toxin from Guam, BMAA, that's killing the people down there, and two isomers of BMAA called DAB, 2,4-diaminobutyric acid, and aminoethylglycine, AG. They're same molecular size, just different arrangement of the atoms, which are even more toxic, and people are breathing this every day. So we're a little bit worried about this. We know what happened in Owens Lake and how that messed up people's health. We're trying to hope that we can be part of the solution to try refill that lake. A professor of neurology at Dartmouth, Professor Elijah Stommel, read our papers. He started mapping on Google Earth the residences of his ALS patients. Found they're all clustered around these lakes in New England that have cyanobacterial blooms. And you can live, if you live within a kilometer of one of those lakes, your risk of getting ALS goes up 25-fold. Now, fortunately, ALS is a very rare disease, but still, we're worried about this. We started getting really worried about Lake Okeechobee down in the center of Florida. Lake Okeechobee has so much agricultural input of nutrients and sewage. There's these big mass outbreaks of cyanobacteria, and then the Corps of Engineers was releasing them down the St. Lucie River to Stewart Indian River Lagoon on the east coast, and down the Caloosahatchee, down by where Fort Myer is, and Naples. On the west coast, physicians started reporting cases of people coming in. So we've been analyzing, and it's just out of control there. We've been quietly advising the Corps of Engineers to stop doing this. But it really worried me because I was up collecting in Lake Okeechobee these samples, and in fact, the day we took a picture of my glove there in the water, and some local people came up and said, "Why are you wearing all that stuff?" I was wearing hazmat, and I said, "Well, this is really dangerous." Well, they had their kids out swimming, fishing, picnicking. They said, well, nobody told us. If you live down in Stewart, you got these big warnings on the beach, but people forgot about Lake Okeechobee. So we're monitoring that pretty carefully. And just to make your evening so wonderful, you get the same stuff here in Lake Tahoe. Sudeep Chandra and Flavia Triboni have done some beautiful work on algal blooms here in Tahoe. I walked along the lake today. I thought it was great. But you need to be careful. You need to be careful. And these problems can be fixed just by stopping the pollution. And it's my silly view, folks, that everybody in this country deserves, well, everybody everywhere, but particularly in the United States, deserves to have clean water. In fact, it's the law. And so count me in supporting your efforts to keep this beautiful lake here clean and free of this stuff. How does this work? Well, the neurotoxin BMAA gets in and replaces L-serine in proteins and causes them to misfold and get messed up. So I wanted to find a place where people didn't have any record of Alzheimer's or ALS at all. This took me to that little village you saw in the CNN report, Ogami Village, a remarkable place. I sat with 8 of these women. They lived to well over 100 years old. There's some 111 out chopping wood. They had never even heard of Alzheimer's disease. When I explained in our country that everybody's scared of it because it hits you when you're old and destroys your memories, they were horrified. They couldn't believe anything like that. We analyzed all their diet at our lab in Jackson Hole and discover they're getting 4 times the amount of L-serine as we are. So that's what led to our phase 2. It's an advanced FDA-approved clinical trial at Houston Methodist for 100 patients. Nobody, the physicians and the patients don't know who's getting what. Some of them are getting serine, some are getting placebo, and we'll know certainly by this time next year if it works. And I said that because I'm hoping I can get invited back here. This is a pretty nice place. Okay. But let me show you what these people are like. This is this lady, and she's typical. So this is not extraordinary. Oops, go back here. She's 98. And what I want you to look at here is not just her range of motion, but her fluidity of motion. These women that are 100 years old, they move like ballerinas. They have absolute recall to when they're 3 and 4 years old. I asked her, can you touch your knees? She puts her hands flat on the tatami mat. I can't even do that. And then I want to see if I've got an acrobat. Would you lean backwards? That wasn't such a good idea, she says. She is 98 after all. But then I want to see, you have a neuron that goes one cell from your spine to your ankle. I want to see her leg motion. I ask her to lift her legs up. Look at this. Bam, 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 bam. Amazing. And they're getting their serine from tofu. In fact, I will say one thing. I've given up popcorn and I'm eating edamame watching movies now. But wouldn't it be amazing if this turns out to be true? We don't know yet if it works. We know it works in monkeys. And if you got a monkey with Alzheimer's, you bring it right to Wyoming, we know how to treat it, okay? But moving that to the clinic, we're waiting on the results, we don't know. But wouldn't it be cool if just by something as simple as tweaking our diet, we could reduce our risk? So these are some of the things that are happening with the Hitchcock Center for Chemical Ecology. I think you're going to have such a great ride with this team. You got the right people, the right science, the right questions. All the stars are lined up. And I'm just so honored to be part of this, to be invited. Thank you so much. Professor Jeffers invited me, and thanks all of you for your patience with me. And if you get interested, we send out a newsletter every month. There's one here on how to create your own blue zone. There's a few up there. But if you come up to Jackson Hole and tell me that you were here from Tahoe, Why, if I have a sandwich, I give you half of it, okay? Be welcome to show you around our lab. And let's get behind this team here at the University of Nevada, Reno. Thank you very much.
Thank you. Thank you so much. Said it's such an honor, and as usual, an amazing talking environment. He's happy to take any questions before we migrate outside, and we'll be wandering around. So if you have any questions about his work, he'll be out there. And any questions about chemical ecology, I could point that I know about that as well, or the center, we listening. But if anyone has any questions for Paul, now's a great time. Please raise your hand. Kelly.
Yeah. First, thank you for this research. Thank you so much for this research that you're doing. It's phenomenal. So being summer in California, the California Delta is 3,000 miles of waterway that jumps into, you could go really anywhere in the world from there. It goes into the Golden Gate Bridge. So where we are, it's a little community called Discovery Bay, and we have blue-green algae. And so it's interesting you say it's lawful that we all have clean water. Is that drinking water that's lawful that we have to have, or?
I wish you could have been with me in Okeechobee when we're collecting these cyanobacterial samples, because we'd go have our lunch at a hamburger joint, had bags of the white powder. I'm not going to tell you what it was, but you might guess. Pouring it into milkshakes and drinking it as much as we can. I think the best thing to do is please stay away from cyanobacteria. Just stay away from it, okay? So if you're out there swimming in green gunk, then don't do it, okay? Until we say it's safe, but I don't think that's going to happen, okay? And why do you want to go to California anyway? I mean, Nevada is where the secrets of the world are. Yeah, there we go. Okay. Thank you.
Pretty.
How far away is California from here? Is it a long way?
So, do all blue zones have the same level of L-serine and red dye?
That's a great question, and the answer is I don't know. We do know that if you adopt the Mediterranean diet, which is rich in vegetables and fruits, olive oil particularly, a little wine, that your risk of Alzheimer's goes down. And we know that through a beautiful clinical trial done in Sydney, Australia, where they put some people on the Mediterranean diet. The others were still eating 4 and 20 pies and ketchup and a beer in one hand. And sorry, Mick, but the diet that you and I love is not as good as what the Italianos came up with. Okay?
So, what other question?
There's one back there. Back there.
Yeah. It's like a...
No, let's put it in the microphone so everybody can hear.
Thank you again, Paul. Can you remind us what ENAA is? And is it found? Have you sampled drinking water? And what's the fate of it?
Yeah, it's beta-methylamino L-alanine. Alanine is one of the 20 amino acids that makes our body up. Cyanobacteria produce a version of that amino acid which has a secondary amine group, and then it's methylated. And what that means is it's highly reactive. So fortunately, as far as we can tell, probably most good water treatment plants would take it out. But if you've got green goo coming out of that tap, or you looked at the dock and it's all green, and you're there in this beautiful part of California and just want to jump into that green goo, please remember what I'm telling you. Don't do it. Stay away. Yeah. The question is, why do they produce it? Because as far as we can tell, all the species we've studied can produce. And one of the ideas is it might be a quorum signal that cyanobacteria use to coordinate their activities. That was an idea from Professor Birgitta Bergman at Stockholm University.
Also, we talk mentioned. So we need all the toxin gets by accumulating up the food chain.
Correct.
But cyanobacteria are also eating many water bodies.
Correct.
Zoom let them eat by fish, I went instead. There evidence that this bioaccumulation that happened in Guam happens in other.
Yeah, well, first of all, if you're still eating bats, we're going to hold a emergency meeting of Bat Eaters Anonymous. But work done by Larry Brand at the Rosenthal School of Oceanography in Miami, and our colleagues Birgitta Bergman and others at Stockholm University shows that it accumulates in shellfish. And it's really interesting. I guess the idea is that it drops as it dies down to the benthos, and like an oyster is filtering maybe half a liter an hour of water, so it accumulates the toxin. So William Camus in the Department of Neurology, University of Montpellier, has documented an ALS cluster among oyster farmers down in France in this cyanobacterially polluted thing. So it's very interesting to me that the Ogemi villagers are very careful what they eat, and they won't eat shellfish. So it's different than what you think, because I would have thought it'd be the top predators, No, like Ciguatera. No, it's not. It's these shellfish. The only thing I must confess to you is I have forbidden my science, my laboratory, from ever testing a Maine lobster. Because I just don't want to know.
So, is there one more question?
One over here, it looks like, right over here.
No. Try to make that EMS problem. And can we stick around after on this for a bit?
One thing I want to know is, do all real genius chemists wear flip-flops? Because I love...
Only it's the stuff.
Only in the summer. Okay, please.
I'm curious that you talked about, I can't remember where it was, the location where people who lived, a kilometer of,
Within a kilometer of cyanobacterial contaminated
lifetime fold.
That's right.
What, was there a long-term, algal blooms that were?
Yes, they're always there, and they're always produced by pollution. And the way to fight this is just clean the water up. Ditto with Lake Okeechobee. But let me put those numbers in perspective. Your chance of getting the incidence rate of ALS in the United States population is about 2 per 100,000. So even if you introduce it 25-fold, that's 50 per 100,000, which is almost the chance you have of winning at one of the tables in the Hyatt Casino. So not very high. So it's nothing to be scared of, but it's something to be aware of, because we can protect ourselves by just staying away from the cyanobacteria. Okay, thank you so much for being with me, and I'm around later if you want to chat, and there's some. Thank you. And thanks to Mick for what he's doing.